So I had a question from a student about the receptors and effectors and things like that, its relationship to negative feedback. So I want to just briefly address that. So remember, we have receptors. These can be free nerve endings, which would be dendrites, which we haven't talked about neuron anatomy yet, but free nerve endings, endings, or dendrites. They can also be specialized structures connected to neurons. So what these are going to do, and I'll just kind of draw one here. So that's a specialized sensory neuron. Signal is going to be going this way. So from the receptor, it's going to synapse with another neuron. And we'll get into the different pathways later, but this is ultimately the basic pathway. We have the control center. And then we have the control center sending out a signal to the effector. Okay? So, receptor, control center, and effector. So the receptor detects the change. The control center decides what to do. And the effector carries out the response. Now, let me address this. Negative feedback. So in negative feedback, here's our set point. With negative feedback, we see our dynamic equilibrium. All of a sudden we see this, way above the baseline. And if that's abnormal, then we need to make sure it goes back to the baseline. So negative feedback would negate the current effect. It would have the opposite effect. And so it would bring that parameter back to the baseline. Positive feedback would be this. So we get a change, and then something happens, and that change triggers more of the same change. That continues to trigger more of the same change. That's actually positive feedback. Now, regardless if we are doing negative feedback or positive feedback, there still is receptors involved. There's still a control center, and there's still an effector. So, receptor, control center, effector, this feedback loop isn't just for negative feedback or positive feedback. It's for all feedback. Not just positive and negative, it's for all feedback. So here's our set point. We know that this is dynamic equilibrium. We know that this would be negative feedback. In fact, what's happening here with this dynamic equilibrium is also negative feedback. It's just very, very small fluctuations of homeostasis. We know that this is positive feedback. We get more of the same. So if we were to map out this and relate it to receptors and effectors, I'm just going to do this. We could say this. So here's our dynamic equilibrium. So what we have if I just kind of do this, not perfect, but so what we have is that when it gets above the set point, the receptor detects this change, sends a signal to the control center. The control center decides what to do, sends the signal to the effector. The effector then carries out the response, which then results in this particular case, if we're talking, let's say, heart rate. Now we use body temp, because that's what we've been talking about. Then it will cause mechanisms that bring that body temperature back towards the baseline. But because dynamic equilibrium is these gradual fluctuations all the time, what we're going to see then, the same thing over here, is that when it gets too low, again, we have a receptor, sends a signal to the control center, which decides what to do, sends it to the effector. The effector then is going to initiate the change, and that's going to cause the opposite response. And that continues back and forth, back and forth. Let's look at positive feedback. I'm going to do this a little different. So here's our set point. Here's our dynamic equilibrium. Here's our upward swing. So let's say this is positive feedback. So at this point here, this change is going to send a signal to a receptor, or be detected by a receptor, sends a signal to the control center, decides what to do, sends it to the effector, and in positive feedback, that effector then is going to trigger more of the same response, and so we get more of that. Once it reaches another level, then we get this whole cycle again, and then we ultimately see a further increase in that. If you think about labor contractions, so we get the stretching of the uterus smooth muscle, of the uterine smooth muscle by the baby. That triggers receptors. They send a signal to the brain, specifically the hypothalamus, which then also acts as the effector, and it releases oxytocin. That then, which is released, then binds to receptors on the smooth muscle of the uterus, which triggers contraction of the uterus, which starts to move the baby out. Well, the movement of that baby out causes additional stretch on the uterine smooth muscle. It activates those receptors, sends a signal to the hypothalamus, releases more oxytocin, which binds to receptors on the smooth muscle, which signals more contraction of that uterine muscle. So regardless if we have negative feedback or positive feedback, we're still going to have receptors, the control center, and the effector playing a role. It's just what is the outcome? Is it going to have the opposite effect of what originally took place, or is it going to cause more of the same effect? Hopefully that helps explain things. Hopefully that helps explain things a little bit better for everyone.